Can everybody hear me okay?
Yeah, loud and clear.
Great. So before I actually begin with my talk, I really just wanted to show this slide to say a really massive thank you to all of you guys that have donated your your urine, and therefore making the study possible that I'm about to kind of talk to you about today. And although we haven't gotten as far as we would have liked, for all the obvious reasons, you know, we've been through various cycles of lockdown in the last year, I am happy to say that I've got some results that I can share with you today. So this is the slide that I started with a few years ago, how your wee can help us understand BBS. And now I'm changing it to how your wee is helping us understand BBS. So the other thing that you might have noticed is that the other thing that changed on the slides was the logo. So I'm going to start off this talk with some good news, and it's that our research team has actually moved, and we're still at the University of Mainz. We're still on campus, but we're now in a very, very fancy new building. So what you see on this slide are two pictures of buildings, and we used to be in this old building on the left, which, whilst it looks quite nice on the outside, I can promise you that inside it wasn't as nice as you'd expect. Our labs were in the basement. My poor students had to share their rooms with cockroaches and spiders, and we were all over the place. So we had lots of different rooms, lots of walking from one place to the next. And in March this year, we got a swanky new building that had just opened, which is the building that you see on the right. And inside the new building, we now have fancy new labs. So on this slide, I just wanted to show you some pictures from our lab. So there's 3 different pictures. And in each picture, you've got one of the people in my team working hard at the bench, all wearing masks and all socially distanced, because obviously that's the new working requirements these days. But I could definitely say that having new building and the new equipment and the new lab where we're closer together has really made a difference, I think, to the kind of working environment. And I definitely think it's motivated us more. And it's nice that we're closer together. I'm closer to my students, which I think is great. I don't know how they feel about it. And we're all closer to the coffee machine, which is a very, very important thing when it comes in a scientific research lab. So with that being said, I wanted to remind everyone what it is that we've been doing. And the project that I'm going to talk to you today is very much just one of the many projects that we have going on in our lab. But as a reminder, you all know that our body is made up of many, many different cells. And these cells come in all sorts of shapes and sizes. So the cells sometimes come together to make organs and tissues. So, for example, we have cells in our kidney, we have bone cells, we have blood cells, we have muscle cells and skin cells. And they all look very different because obviously they have different roles and they do different things. But some cells, and in fact, a lot of the cells, have this little kind of antenna that sticks out of them, out of the surface. So on this slide on the left, you just have kind of your classic view of a cell, which is kind of a round blob of a larger circle in the middle, which is the nucleus. And if we look on the surface of these cells, which is what we're viewing on the right in the black and white image, I hope some of you, you might be able to see it. If not, I'll describe it. Some of these cells have what looks like a little kind of hair or kind of a little radio antenna that's actually sticking out of the surface of these cells. And it's these little hairs, they're called cilia. And I'm sure many of you have heard of this term before, because it's these cilia that actually don't function very well in BBS patients. And we are just trying to figure out exactly what these cilia actually do. And it really was work that Phil Beales started, you know, over 20 years ago that helped to identify the role that these cilia play in many different diseases, and how important the BBS proteins are in the cilium functioning. Now here we have just a cartoon of one of these cilia, what it looks like. There are lots of complicated parts to it. I don't want you to remember any of those. The only thing I want you to remember is the Wi-Fi signals coming out to the top of the cilium. Now, the reason for that is that people used to think that these cilia were just functioning to receive signals, so getting signals, kind of receiving signals. And that was already a very great finding. However, more recently, we now know that cilia are not just able to receive signals, but they can also send out signals and send out kind of little parcels that contain some kind of molecules inside them that might be doing something, might not. So what you see now is that same picture and that same cartoon of a cilium, but instead of the Wi-Fi signals at the top, you've got kind of small little blobs and circles that I've kind of depicted as packages and parcels that are actually being sent out and from the cilium. Now, in the scientific language, these parcels are called vesicles, extracellular vesicles. And scientists in other research fields are looking into these parcels and vesicles and trying to figure out what they do. And what we found out is that their role is actually to help cells communicate. So cells can talk to each other via kind of sending each other parcels. So I kind of like this diagram that I've showed up on the slide here. There's 3 cells next to each other, and the cells are kind of talking to each other. So the one on the left and the one in the middle are kind of high-fiving each other and saying hi, whereas the cell on the right, yeah, that's right, the one on the other end and the middle, they seem to be more like, you know, annoyed with each other. The point of this slide is to say that this communication between cells can sometimes be done via these parcels. But the problem is we need to figure out what's in these parcels before we know what these cells are trying to say to each other. So that's kind of what we've started looking at, because we thought it was very, very interesting, because we thought that these parcels might be different if they come from a BBS patient versus someone that doesn't have BBS. So we needed to find a good way to catch and look at these parcels. And I think I reminded you before that we can actually catch these parcels because they actually end up in your urine sometimes. So many of you know that your kidney is important for making urine. And in your kidney, the urine flows through lots of tubes, and those tubes are lined with cilia. And as the urine flows through these tubes, some of these small parcels that get released from the cilia and the ciliated cells can end up in the urine. And so what this means is that we as scientists can collect the urine, and we can use fancy preparations in the lab to isolate these parcels. And just very quickly, the way we do that is we collect the urine, we put it in a tube, we spin it down for a very long time in a very, very, very fast centrifuge, and then we kind of collect what we get at the end. And this is the last slide that I showed you last time, which is that we'd shown that this was possible to do. And what we get at the end, if we look at it on a very, 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 very high resolution microscope, we can actually see these parcels. So what you're seeing here are 3 pictures of these parcels. And, you know, they really are tiny little blobs that look a little bit like deflated footballs. But they show us that we've got the right structures, and we're able to isolate them, and we can try and have a look at what's in them. So the questions we asked last time was, whether people with BBS have different contents in these parcels. And the ultimate question we really want to know is that if this could be why the kidneys in BBS patients might not work very well. So the way we study this is that we can isolate these little parcels and isolate them, and we can actually take pictures of what's inside them. Now, the technical term for this is actually called a Western blot, where you're isolating the protein and looking at it on a kind of membrane. But to be able to explain this to all of you, all I want you to remember is that if we take the parcels and clean them up from one patient, and if we get a big fat band, like we see here on the left, a big fat band, if it's dark, it means we've got more of a protein. And then in the middle kind of grey picture here, you'll see that the band is lighter and it's weaker. And that means we have less of a protein. And at the very end, there's a person and the picture that came from her samples, there's nothing there. And if this was an interactive thing, I'd ask you to guess what that means. Well, it means that if there's no band, it means that we don't have any protein there. So this is the way that we can look at certain types of proteins. And the first thing we did when we started working with the urine that we'd collected was trying to find different ways of cleaning up the parcels. So this is kind of a photograph, and on it we've got lots of different bands, and that they're different methods. So we've tried different methods of cleaning up these proteins. And I hope that for some of you that might be able to see the screen, in one of these trials, we have a really, really thick band. That's the one with the triangle. So after lots of kind of playing around, trial and error, we found the best way to clean up these parcels and to take better pictures. So once we'd optimized that, we were able to actually start the real work. And one of the first results that I can say today is that we found a protein that seems to be quite interesting. So if we look at the control group of people, it seems that the amount of protein, so the kind of intensity of this band, seems to be more consistent. So this is kind of depicted by the two pictures on the left. So the two bands from the control people seem to be relatively the same. And what we noticed that in people with BBS, the amount of this protein seemed to vary quite a lot. Sometimes we had darker bands, as you can see in one of these pictures here. Sometimes we had lighter bands, as you can kind of see at the end. And sometimes we had no bands at all. So that was something that we thought might be interesting. That being said, we don't have enough controls, so it might be that this is something we need to obviously repeat. The second result that I can show you today is that even when we took samples from the same person, so this was actually a BBS patient here in Germany who lives about 2 hours drive from here and is a fantastic donator of his urine. And he's very, very excited to be part of this study as well. So we found that when we get urine from him, and we see that even with him, we do see variations over time, which makes sense because we realize that a lot of this, what your picture looks like of this protein can depend on how much you've eaten or how much you've had to drink. So we still need to find ways to kind of adjust the results and adjust the experiments to see that we have a good normalization method. But what has been amazing to see is that we can store this urine and freeze it and then thaw it 2 years later, and it's still good to work with. So we still are able to get valuable and useful information out of this. Now, the last result that I want to show you and share with you today is, I think, the result that we're more excited about. Because it turns out that these parcels, when you isolate them, and if you put them on a special kind of cell, so that's depicted here on the left. So we've taken these parcels and we use kind of a pipette dropper, and we put them onto a special kind of cell that it's called a reporter cell. So what that means is that if they are responding to some kind of treatment, they glow and they emit light. So that's depicted here by these sunbursts that are kind of glowing up in this reporter cell. And we can measure how much a cell glows depending on how much light it emits. So what we saw was that, again, despite the fact that this urine was sometimes stored, frozen and stored in the freezer for a couple of years, we're still able to see a response. So the graph on the right is basically just a measure of that response. And the high tall graph is actually the positive control. And the one at the end where it says serum starve, that's kind of a negative control. And the 3 bar charts kind of in the middle, or the bars on the left, so the first 3, those are actually, that's the response we're getting after we treat these reporter cells with these parcels from the urine that's been treated in different ways. So at the beginning, we're just trying to optimize the assay and to figure out, if this is at all possible. So that was kind of a really exciting finding, to see that these cells, these parcels, do actually do something. So like I said, I very much wish that I could update you more. I wish I had more results to show you, but we ourselves have been in, like I mentioned, various rounds of lockdown. And as I'm sure you can imagine, even just these 3 slides of results, there's many hours and days of data collection that lie behind it. So there's a long list of things that we need to do next. So first of all, we absolutely need more urine. The more we have, the better. Because as you saw, we saw that there are changes even in the same person over time. We also need to find better controls. So that's been one of our biggest challenges, because what we actually need, at first we collected all the urine from parents and siblings, but we're realizing that that's not very helpful. What we actually need is age and gender matched controls. So that's kind of the biggest challenge, is to find age and gender matched controls to the samples we've collected. We also want to try and match the type of BBS mutation to the amount of protein that we found, that can sometimes be different, because as you're all aware, there's many different BBS genes with different mutations and different proteins. So that's something we want to, if we ideally had results from everybody, that would be amazing. And lastly, we're very keen to do more experiments to figure out what these parcels, the response of these parcels on our reporter cells. We've got some preliminary data that I didn't want to show here because we've only done it a couple of times. But we think that the parcels from a BBS patient might actually respond a little bit different to one of these parcels from a gender and age match control. So that's where we are. In a nutshell, I need to say we are going to need more we. So here I have my little kidney with a microphone as a cartoon, and it's saying, thank you so much. Your we is like liquid gold to us. And I can't wait to come next year to collect more. And I'm grateful that anybody is willing to donate their we. And so with that, I'm at my last slide. I want to say a massive thank you to all of you guys. I also want to say a massive thanks to my team who make work, especially through the pandemic, a really great place to be. So especially the people who've worked really hard on this has been Ann-Katrin and Alina, and And I'm pleased to say that despite the pandemic, we've still managed to recruit some more students. Now we have Rike and Vanessa, who are some new students that have started. And as I say, every year, we're totally happy and excited to have people come and visit our lab if you want to have a tour and see what we do. I know a couple of you have taken us up on it. And now that we have this fancy new building, we're more excited than ever to kind of share what we do, share the research we do, and show you around.